Good day to everyone. This is Ziwan from University of Malaya in Malaysia. I will be presenting our work on the comparative analysis of transcriptional regulation in tropical and polar green algae under heat stress. This study is part of a collaborative project between and the National Taiwan Ocean University on understanding the underlying mechanisms of algal responses to climate changes, which include global warming, elevation in UV radiation, and ocean acidification. This graph compares the current rate of warming in relation to the end of the 19th century. In 2017, human-induced warming reached approximately 1 degree Celsius above pre-industrial levels. At the present rate, global temperatures are expected to reach 1.5 degrees Celsius by the year 2040. We all know that the polar regions receive less intense sunlight compared to the equatorial region, which experience a tropical climate. This is because of the longer distance from the sun, and the solar energy is spread out over larger areas near the poles. However, the warming phenomenon does not only affect the tropical regions, but warming events are taking place across the latitudes, as shown here in this diagram from the National Oceanic and Atmospheric Administration, or NOAA. As phycologists, we are interested in the effects of climate change to the algae. The phytoplanktons or microalgae play an important role in the ecosystem as they are one of the major primary producers in the food web. Increased warming or other shifts in environmental parameters can affect the size and composition of the algal communities, which then leads to profound impacts, including changes to nutrient cycling and a chain effect to each of the trophic levels. We humans at the top of the food chain stands to lose the most, with the obvious being our fisheries productivity. Other possible implications on ecology include the shift of species range and distribution, where we can see warm water species expanding their range either northwards in the northern hemisphere or southwards in the southern hemisphere, or the loss of biodiversity with regards to the stenothermic organisms. There are also increasing reports of harmful algal blooms intensifying with climate change, and this is a serious concern because these algae produce toxin which renders seafood to be unsafe for human consumption, and the eutrophication also causes low dissolved oxygen in the water, thus killing fishes in the aquaculture farms. Climate change also indirectly affects the nutritional quality and taste of our fisheries product, as they feed on the algae or phytoplankton. I would like to add that microalgae are good proxy for climate studies as they are sensitive to changes. They have relatively short generation time, and most are not exploited commercially in nature. Moving on, the current study is derived from an earlier study where we sequenced the whole transcriptome of an Antarctic chlorella strain subjected to its ambient and heat stress temperatures of 4 degrees Celsius and 33 degrees Celsius respectively. The heat treatment was conducted for 120 hours or 5 days before total RNA was harvested. Based on the transcriptome data from that study, we designed 40 targeted probes to detect expression of genes that are potentially differentially regulated in an Arctic and tropical chlorella strains to investigate their molecular response to heat stress. The following two slides are meant to show the list of DEG obtained in the earlier study. And the current slide shows the 40 custom-designed nanostrain gene probes based on the list shown a moment ago. This includes 3 housekeeping genes for normalization. Before I talk about the experimental design of this study, I would like to highlight some of the related studies that have been reported. This study by Huang et al. Uses cDNA microarray to analyze the transcriptome response of an Antarctic diatom, Chitoceros neogracili. They transferred the cells from 4 degrees Celsius to 10 degrees Celsius for short durations of 15 to 16 minutes and 3 days. They found that the expression of most genes were largely affected within the first 60 minutes, and differential expression was no longer detected by the third day. This is a diagram from the SAID study showing the time course dependent fold changes in RNA profiles from 15 minutes to 3 days of response to thermal stress. Next is a study conducted on a tropical cyanobacteria subjected to high temperature treatment of 42 degrees Celsius as compared to its ambient temperature of 35 degrees Celsius. And RNA was sampled at different time points to compare the immediate and long-term or acclimation responses. Here we can see a different trend of expression in the immediate versus long-term response. In general, there are two major approaches to study transcriptional responses, either using RNA-seq, which can detect far more transcripts, SNPs, or mutations, as it does not require species or transcript-specific probes. On the other hand, microarray, qPCR, and the nanostring encounter approach that we are currently using requires known sequences for probe design. The nanostring technology that we are using, however, does not require any amplification. Right, in the current study, we've selected an Arctic and a tropical chlorella strain and narrowed down the range of stress-inducing temperatures based on our previous publications. For the polar strains, they generally show stress response at above 30 degrees Celsius, while the tropical strains are heat tolerant up to 40 degrees Celsius. So we incubated the selected strains at the temperatures shown in the table at the top left corner to identify a suitable stress-inducing temperature by monitoring their growth and photosynthetic parameters up to 10 days. For the Arctic strain, it could not survive at 38 degrees Celsius. Therefore, we chose 35 degrees Celsius as the stress-inducing temperature for the Arctic strain, while 40 degrees Celsius was chosen for the tropical strain. We then identified the suitable RNA harvesting time points based on existing literature, which are at the 2nd and 6th hours, while the last time point was established based on the decline of the photosynthetic FVFM parameter down to half of its value under control condition. This graph shows the photosynthetic maximum quantum yield of the Arctic strain measured using a water PAM fluorometer at 4 degrees and 35 degrees Celsius. The red circles indicate RNA harvesting time points. This is for the tropical chlorella strain, which is incubated at 28 against 40 degrees Celsius. The following two slides show the table of 8 differentially expressed genes that showed obvious signals between the two strains. These genes are generally involved in antioxidative stress response, carbon metabolism, nucleotide metabolism, biosynthesis of amino acid, and protein processing in the endoplasmic reticulum. Unfortunately, most of the other genes did not show much signals, and I will offer a possible explanation in a short while. This graph shows the total counts of the genes detected by the custom design probes for the polar strain across a time series when exposed to 35 degrees Celsius. The data shown are mean plus minus standard deviation of 3 replicates each. In general, most of the gene regulations under heat stress are more actively expressed during the first few hours as compared to after several days. As for the tropical strain, we detected much less signals of the target probes, but the expression of carbon metabolism genes appeared to be going up back to normal after the initial repression of expression, probably a result of acclimation to the heat treatment. I have put together some of the points to consider when we interpret the gene expression data from this study. First, although we confirm that both strains used were species of chlorella, the target probes designed based on the Antarctic strain from a previous study may be too specific, and thus they were not picked up by the strains in the current study. The lower signals observed for the tropical strain may also indicate incomplete hybridization of the probes, although we followed the manufacturer's recommended protocol. Varying degrees of susceptibility to stress have been reported among different species, even if they are from the same genus. Expression patterns may even differ between biological replicates, as seen in the large standard deviation from the graph shown earlier. Next is that genes putatively encoding proteins with the same function can be simultaneously up and down regulated. The lack of differential expression seen for some of the genes may be due to post-transcriptional control. This is to show the various transcriptional patterns of genes reported in a study on tropical cyanobacteria. For instance, graph A shows a rapid increase at the beginning, which then levels off with time. But in graph C, the genes decrease at the start and then return to normal. To summarize, due to the relatively small number of green algal genomes available and that they are incompletely annotated, it is rather challenging to design target probes that are suitable across taxa at this stage. The general responses to heat stress as observed in this study include regulation of nucleotide metabolism, which involve the synthesis and degradation of nucleotides for cellular division and maintenance. Second is the regulation of protein synthesis and degradation to replace those which are damaged due to heat stress. Third, regulation of carbon metabolism to resynthesize more or conserve existing energy resources. And not the least, anti-oxidative mechanisms being activated to counter the stress. And that's all from me for today, and I hope you enjoyed the presentation. Thank you.